So hello and welcome to the International IVF Initiative, and this session is entitled Personal Genetics. So this is really something that we are really passionate about, and it's actually just the forefront of what we're going to start to feel is just a natural progression of technology, a natural progression of medicine and embryology. So thanks everyone for joining. We'll be starting momentarily. I am joined by, and I'm honored to be joined by Sissy, Dr. Sissy Wamaitha. Dr. Sissy, do you want to introduce your background and your bio?
I will do. So I'm a stem cell biologist by training, and I've worked on reproductive biology and early human embryos. And I'm currently researching how gametes are formed and how we can use our basic research understanding to translate these to infertility therapies and improving the options that people have, whether or not they can make their own functional gametes. So I think you're going to talk a bit about in vitro gametogenesis as well, or maybe John is. So very much in my wheelhouse.
Great. It's an honor to have you. You were definitely hand chosen as just a preeminent expert in this area. So it's really great to have you on board. Just to let everyone know, for videos of previous sessions, please go to the ivfmeeting.com. You'll also find information about upcoming sessions on that page. Thanks for joining us tonight. My name is Dr. Helen O'Neill, and I am co-moderating this session tonight. So as we mentioned, we encourage you to ask your questions using the Q&A at the conference service, not the chat function. Okay, so I think we should just get started.
I think we should. I think what I'll do as repayment for your wonderful introduction, Helen, I will introduce you, given that you are our first speaker and will be opening the session. So Dr. Helen O'Neill is a geneticist and reproductive biologist whose research focuses on early embryo development and uses gene editing to improve our understanding and treatment of infertility disorders. I've known Helen for some time. She's an excellent scientist, and I'm very excited for her to open today's session with her talk on CRISPR genome editing, how, what, and why. So Helen, please.
Thank you. Let me just share my... It started in the middle of my slides, so I'll start at the beginning. So what I'm really excited to talk to you about tonight is actually not even a new technology. It has been around for a number of years now. It's still considered a new technology. Hi guys, it's saying that screen sharing is not enabled. Could you help me with this? Okay, we're in. Great. So I'm here to talk to you about genome editing, how, what, and why. And I feel really passionately about this area, not just because of its applications in medicine or in genetic understanding, but because of its applications among every single field we know. So I would love to tell you a little bit about how it works and where we might see its application in future. So my disclosure, I am CEO and founder of Fertility Health, but there is no conflict of interest there with this. So CRISPR genome editing has been referred to as genetic surgery, molecular scissors, a find and replace tool, and it is all of these things. It has become a preeminent tool in our ability to not only make edits, as the name aptly suggests, but also
Helen, I'm going to stop you briefly because we can't see your slides. You can't see your slides. Do you mind sharing them? Thank you.
Sorry, I thought I was... Technology. The guys will kill me for not having done... So it did say I was... Okay, tell me when they're... Are they now sharing?
Perfect, we've got you.
Okay, we have. I won't repeat myself. Just to go through my disclosure, and this is about where I had gotten to, so you haven't missed anything. So these these terms that we use to explain genome editing go a long way to describe its applications. Talking about it as genetic surgery is a really great way of understanding how it could be used essentially as a very fine tool. When you look at some of the current tools that we use, actually surgery probably doesn't go a long way in saying how accurate it could be on a molecular level. So maybe using the word molecular scissors or a find and replace tool are a little bit more appropriate, but you will see these expressions used interchangeably in the mainstream media. So since its initial kind of seminal paper in 2013, this has dominated the headlines for not just Nature and Science, but also global headlines in terms of the mainstream media. And it's no real surprise. It has really revolutionized many of our ways that we can adapt, many of the ways that we can understand and appreciate biology. So you can see really quickly from this diagram here, or from this graph, that since the seminal paper in 2013, there has been an explosion. This actually doesn't show the full appreciation. There are now over 20 million search hits for the term CRISPR when you Google CRISPR. So what I'm going to try and explain to you tonight is essentially a snapshot of the technology where it is now, but it is really like trying to explain trying to take a picture of somebody who is running. You get an overall impression that what is happening, and you know they're going fast, but truthfully, this technology is moving so quickly that even by the month, some of the techniques are becoming outdated. So I really think that's important to remember. Even in the 1 year following the seminal paper by Emmanuel Charpentier and Jennifer Doudna, where they applied CRISPR to genome engineering, it has been used in almost every other model organism there is. And I always think that this is a real testament to a technology when it is easily replicated and reproduced, and results are successful in the hands of any scientist. And this is certainly the case with CRISPR. But what is CRISPR? People often think that this is a new, strange, or foreign phenomenon, and it's a new technology. And yes, it is a technology, but really what this technology is harnessing a very simple, very old technique, which is a bacterial immune system. So CRISPR itself is not a new thing. CRISPR is an ancient immune system used by bacteria in order to fight off invading viruses or other bacteria. So it's actually quite nice to look at the process here. We often remove ourselves of the physical entity of what is happening, but this picture shows a phage landing on the surface of a bacterium. And as you can see from this phage, it's actually physically inserting its DNA into the bacterium. You can see it down here. And this is an electron microscope photo of this happening. And what's wonderful about this, as you can see, the phage inserts its DNA and then just flies off. What's wonderful about this is the fact that when you understand that actually a virus, once it inserts its DNA into the bacteria actually has an immune system that fights this off. And that immune system is an adaptive immune system called CRISPR. So it would be remiss of me not to explain what the acronym is. It stands for Clustered Regularly Interspaced Short Palindromic Repeats. And essentially what CRISPR refers to is the genetic sequence in this bacterial genome. So this can be very easily broken down and be easily understood if you actually just say the words a little bit more slowly. These regularly interspaced short palindromic repeats were found clustered throughout the bacterial genomes. And they were very elusive in that scientists didn't understand what these sequences were and what was the interspaced sequences in between. What we've uncovered is that these interspaced sequences actually served as a genetic historical timeline of invading bacterial genomes that had been inserted into a bacteria's genome. In other words, it served as a historical timeline for every time a bacteria had an infection from a virus, fought that infection and won by inserting that piece of foreign DNA, thereby rendering itself immune to any further infections of a bacteria of a similar genetic sequence. So when we talk about CRISPR, we always say CRISPR-Cas, and really the Cas stands for the endonuclease, or what I was saying earlier, the molecular scissors. It's essentially just an enzyme that will cut the DNA. Now, typically, we always refer to CRISPR-Cas9. I always think Cas9 is like the playgirl of the CRISPR world. It was the first endonuclease that was used. She was quite promiscuous, was known for cutting all sorts of places in the genome. But actually, since the discovery of Cas9, hundreds of other bacteria have been sequenced in order to find more accurate, more precise, and just other alternatives for Cas enzymes or endonucleases. And actually, where we're going with this is not just using a standard endonuclease, we're using synthetically altered enzymes that allow us to have a greater packing capacity size-wise. Sometimes Cas9 was too big to get into the cells. So we're really evolving in both our tools, but equally the endonuclease that does the cutting. But it's very important to think of these two components because both of them are very useful in genome editing and beyond. So just to show you a quick example here, we can see the phage landing on the surface of the bacterium. And as it does, as I mentioned, it inserts its DNA into the bacterium. When the bacterium then detects the presence of the virus DNA, it produces two types of short RNA, one which contains the sequence that matches that of the invading virus. And these two RNAs form a complex for the protein Cas9, as you can see this big rock-like sequence in the background. Cas9 is the endonuclease, which is a type of enzyme that can cut DNA. When the matching sequence, known as the guide RNA, finds its target within the viral genome, the Cas9 enzyme cuts the DNA, disabling the virus. Once inside the nucleus, the resulting complex will then lock onto a short sequence known as the PAM. And this Cas will unzip the DNA and match it to its target RNA. So this process here is really, really adaptable to where we can actually not just use this find and replace with these guide RNAs, but also the cutting. As I mentioned, there's two separate processes here that we need to think of. But importantly, as soon as you break DNA, DNA wants to repair. So you'll all be quite familiar with the two types of DNA repair. One is non-homologous end joining, which is very quickly and often referred to as the error-prone method of repair. I think it's probably not as error-prone as we give it credit for. Our cells are constantly repairing, and if it was that error-prone, we'd all be a bit more error-prone. Nonetheless, it is error-prone in the sense that these ends just quickly want to join together, and sometimes you will lose base pairs here and there. Now, for the purpose of an experiment that just wants to knock out a gene, you don't really mind whether or not a few bases are missing. However, if in DNA you were missing a gene, it would be a favorable experiment to do homology-directed repair. So the other type of repair that DNA undergoes is homology-directed. In other words, and this usually happens when cells are dividing, so there's plenty of DNA transcript around for it to enable a correction of that break. So it actually really matters to your editing outcomes what kind of repair you are looking for and what kind of edit you are trying to make. But the idea of doing genome editing with engineered nucleases is not a new one. In fact, there have been many predecessors to this technology that have adapted and tried to use forms of molecular scissors and programmed nucleases, such as engineered meganucleases, zinc finger nucleases, which you'll quite commonly hear about, and transcription activator like effector nucleases. And while these are successful to an extent, they are clunky, and I would essentially call the editing outcomes comparable to what you would achieve on a typewriter. Yes, you can rewrite code, but if you make a mistake, it's very expensive and clunky to refix. And they have been used in the clinic, and we're seeing more and more cases where companies are releasing their phase 1-2 clinical trials using these former methods of genome editing. And the reason is because they've gone through the phase 1-2 clinical trials. There is not one of those companies that is not adopting CRISPR as a replacement for their forthcoming technologies. Here's an example of where genome editing was used in a young girl called Layla, and she had a type of blood cancer whereby she had no white blood cells left and she had undergone every treatment there was. She was given an experimental license to do genome editing on her, and 5 years later, she is now happy and healthy. So there have been incidences where people have actually used an emergency experimental license on humans for somatic cell therapy, or they have opted for them, such as the man on the right with Hunter syndrome. So a quick run through of how somatic cell therapy works is to treat disease in their somatic cells. So the cells that live and die with them. How this broadly would work is that you would take the cells of the actual sick individual themselves, you would edit them to correct the mutation that they have, and then you would grow these cells up and retransfuse them back into the patient themselves, thereby foregoing any need for immunosuppressants. You reduce the risks of graft versus host disease. You're using the patient's own cells to cure their own condition. Obviously, this is much more applicable to some types of disorders and diseases, especially blood-based disorders and cancers. But preclinical studies in non-human primates have shown a single dose effective use of base editing, which actually showed that they were able to permanently reduce LDL cholesterol in patients who suffer from familial hypercholesteremia. So I really think this is going to make massive advances in the clinic, whereby you can actually do a single dose and it's sufficient enough. And I think threshold is a really important thing to consider. The sufficient enough editing change to enable a recovery in a given patient. Equally, there have been some mouse animal studies that have shown potential for fetal gene therapy, doing in utero gene editing to correct disorders that have been detected in utero, such as spinal muscular atrophy here. But what if you are to try and prevent genetic disease? Among the IVF industry, I think it's quite a familiar idea that you would prevent an aneuploidy, but when you come to prevent genetic disease, it's quite a familiar technology to use pre-implantation genetic diagnosis. However, applying genome editing to prevent genetic disease invokes all sorts of negative connotations. So first of all, before we talk about the application of this technology in human reproduction, I think it's perfectly placed to just take a little bit of a time to look at the current treatments that we use for treatment of genetic diseases, such as pre-implantation tissue typing, pre-implantation genetic screening, diagnosis or PGT-A and PGT-M, mitochondrial replacement therapy, which has been granted a license to be used going forward, or else you have the option of a use of a donor. And actually, almost every one of these, barring the donor, could be replaced through successful genome editing technologies. But we'll discuss the logistics of that. But I think when we look at the image underneath, or more clearly here, if we were to integrate genome editing into assisted reproduction, very often people assume that this is an entirely different field, or that it's a gross aberration of the current practice. And in fact, there's very little difference to what we're currently already doing when we manipulate an embryo by injecting a sperm into it. Possibly that is the exact same time that we would inject the nuclease in order to create the edit. At the exact same thing, we would wait for the embryo to grow. The cells would be tested to ensure that they then did not carry the mutation that you were trying to correct, and then they would either be vitrified or transferred for pregnancy. So when we usually talk about any of this, unfortunately, the dominating headline is always about designer babies. And this is a very unfortunate thing, because it really does take from the unmet need potentially in given cycles, whereby there are no embryos left for transfer, and all of them are shown to be affected. Very often there are claims or cases whereby they show things like height or intelligence, or what you'd call extracurricular changes that you would make, and these are all polygenic and misunderstood entirely, so they would never really be done. It always frustrates me hugely that we do not consider that germline genome editing would not be treated in the exact same way as we treat pre-implantation genetic diagnosis, and the introduction of every single new technology or new disorder that we treat for all has to go through the human fertilization, in the UK at least, through the Human Fertilization and Embryology Authority before it is approved. So every single condition is approved on a case-by-case basis. So the idea of this was an idea up until 2 years ago at the Second International Summit on Human Genome Editing, where the news was released that a researcher, He Jiankui, had in fact not just taken his experiments, or had in fact taken his experiments beyond experiments and into the clinic. Through a series of experiments, he had edited the embryos of a number of patients. He had recruited them to tell them that he was coming up with a vaccine for HIV. And what his plans were, which he did execute, were to edit a gene called CCR5, which when mutated, renders HIV incapable of entering into a cell. So his goal was to make these babies immune to HIV. So when this news broke at the eve of the international summit, there was a lot of backlash. First of all, they wondered whether the first claims were to say that actually he was saying that he did it and that he probably hadn't done it. I think for those of us who knew a little bit more, we thought it wasn't whether he had done it, but it was rather the extent to which he had done it. So what he announced was that 2 twin girls had been, or rather one pair of twin girls had been born with their genomes edited to mutate CCR5. There were all sorts of wrong things with this experiment. As much as I am talking very positively about the use of the technology at some stage, there were so many things wrong with this that it makes a lot of us angry for the backset that he's put in terms of other people's ability to use this technology just for research alone because of the major backlash internationally. First and foremost, he did not address an unmet medical need when he was editing a human embryo. There are plenty of antiretroviral therapies for HIV. The actual editing he did was very poorly executed. They weren't actually able to tell following the experiments whether or not they were heterozygous or whether or not they could actually contract HIV still. Nobody's going to expose them. It wasn't actually clear what the new mutations would do. And there were many, many problems with informed consent. So he was pretty much renounced by the scientific community and his peers. And yet there were documents to suggest that potentially his experiments had been funded by the Chinese government. This is him under house arrest. He has actually since been put in prison for these experiments. But it does make you wonder about some of the headlines that came about with the first IVF babies. So you can see from this very damning headline, "Do not work with this man," you would not equate this with the same person who has won a Nobel Prize. And this unfortunately was the premise of He Jiankui's, I guess, motivation, that he assumed that he may be pilloried at first, but that later on he would be rewarded for his work. And I am not in any way comparing the work of these two, but I'm just saying how different headlines can paint a very different picture in different times and scenarios, and they can change. So I think it's very important as a result to put this in context. We have, when you look at both gene therapy and pre-implantation genetic diagnosis, both of them have been attempting to do similar work to what germline genome editing would do. With gene therapy, we've spent over 30 years just trying to ameliorate symptoms or to try and compensate for a protein that is missing by the insertion of viral vectors that can be used as carrier vectors for a given protein. And equally with pre-implantation genetic diagnosis, where it is, as I mentioned, licensed on a case by case, it aims just to avoid. It involves the insertion of needles and the removal of genetic material. It's certainly not free from harm. But if we could, which we can, should we correct to cure? I think it really only takes a quick look at the number of even single gene disorders that affect how many people, 400 million people every year, for us to realize that this is something that actually we really need to address. And the timing is quite right at the moment with it being Rare Disease Day coming up. we are really fortunate tonight. Dr. Wamaitha did her PhD in the laboratory of Kathy Nyakin, who is the first person to have done genome editing on human embryos for research. And we are also joined later by a current member of her lab, who is also going to be talking about their work on editing human embryos. I will say that the fundamental thing that we need to know is how this works and how we can actually start to see whether and where it may go wrong. The initial headlines were to say that the genetic modifications could help researchers develop treatments for infertility, when really, actually, we just wanted to know fundamental information about how an embryo develops. So there have been plenty of safety concerns raised with germline genome editing. And I just want to very quickly say that when you think of a given experiment or outcome, you have to account for every different type of tool that has been used in the CRISPR toolbox. And as I mentioned, all of these tools are evolving by the week. So we have new methods of delivery, new modes of delivery, different types of endonucleases. Our guide designs are evolving quickly. So I think it's only ever giving you one time point and one given experimental outcome. So these are just some of the missing pieces of the CRISPR puzzle, all of which yield different outcomes in a given experiment, depending on what you are using. I call it the nucleotidal wave. It's very difficult to keep up with all of the new advances, but it is certainly exciting. It's really important to know that genome editing is not just being used for editing genomes. We are moving beyond that to the ability to edit the epigenome, to edit our methylation, to do base editing, which doesn't necessarily break the DNA, but actually fixes a single base with high resolution and targeting ability, and also to engineer cancer cells to enable them to overcome the immune system. So the risks of genome editing, I'm being really quickly, Sissy's turned on her camera, which makes me think I'm over time. The risks of genome editing are always that stated that you would have unwanted editing, that you may have off-target effects, that you may get what called CRISPR-derived mosaicism, that not every cell would have the intended correction, or that you may have a transgenerational change. And I just want to take a quick moment to take a comparable look at what the risks of assisted reproduction technologies are. Unwanted editing, I definitely don't think any embryo wants a single cell or a clump of cells to be removed. There have been plenty of papers showing the potential off-target effects of biopsy, of laser, of culture media in general. Mosaicism is the biggest topic and a never-ending drama in the IVF world, and is still consistently debated. And of course, the transgenerational change that we are witnessing one generation down the line after children who have been born from IVF and ICSI. So I think if we were to try and legislate for IVF right now, I think we'd actually really, really struggle. I just wanted to take a note to also have a look at the 1974 paper that James Watson said, where he foresaw all sorts of bad scenarios from IVF and its application. So the limitations are that it's a new technology relatively. Legislation does not move as quickly as technology does. I think there's a lack of human biological material that will enable us to actually do experiments. The only, human trial when it comes to embryos is human trial. And with that, and with an ever evolving technology like this, where it's getting incrementally, in fact, more than incrementally better by the week, it's hard to draw the line on saying when good is good enough, when we don't have enough research in this area. So if you're interested, please look at the Nuffield Council on Bioethics, and I will take your questions and move on. Thank you.
Thank you very much, Helen. Excellent talk as always. I think I've seen you speak a couple of times now. So we're going to go to a question from our panelists. And I'll ask this one briefly, actually, because it's relevant to what you're doing. You've talked about CRISPR, but how many genome editing tools do we have? Is CRISPR a unique case, or are there other ones that have been either previously related to this in genome editing as a whole?
So the previous tools, as I mentioned, were much more clunky tools like talons and zinc finger nucleases. People always joke that talons are the best way to ruin a postdoc's career. They're very time consuming. They're not always accurate. And then the newer tools are using the same principles of CRISPR, but what they're doing is they're using, it's almost being dominated by biochemists who are modulating both the enzymes and the guides. So that's what I mentioned about it being both an ability. The newer tools are actually not just for genome editing, where you can actually disactivate the Cas9 or the nuclease and just use the finding tools. So you can actually use it as more of a diagnostic element rather than a therapeutic element.
Excellent. And we have a number of other questions. And I want to remind all our attendees that we do have a Q&A session after the three speakers. So please continue to submit your questions to the Q&A rather than the chat, and we'll try and get to as many as possible. So our next speaker is Gregorio Alanis-Lobato, who is a computational biologist working with large genomic data sets to study biological problems and to support drug discovery. So he'll be discussing his work investigating unintended targeting effects in his talk on frequent loss of heterozygosity in CRISPR-Cas9 edited early human embryos. Thanks, Gregorio.
Thank you, Sissy. Hi, everyone. So yeah, Sissy said, today I'm going to talk about the pipelines we developed to evaluate the frequency of CRISPR-induced loss of heterozygosity events in early human embryos. So in other words, I would try to make the case that there's still a lot to be learned about CRISPR before we can bring it to the clinic. So I am currently a principal computational biologist for central nervous system diseases at Boehringer Ingelheim, but the work I'm going to talk about was done when I was a postdoc at the Human Embryo and Stem Cell Lab, led by Professor Kathy Niakan at the Francis Crick Institute in London. So as Helen mentioned, the Nobel Prize-winning CRISPR-Cas9 system has become a key molecular biology tool due to its flexibility and robustness compared to other genome editing technologies. CRISPR has enormous therapeutic potential for gene correction if the high fidelity homology-directed repair pathway could be harnessed to insert DNA templates into a locus of interest. But really, the most common use of CRISPR is gene disruption by taking advantage of the error-prone non-homologous enjoining pathway to introduce indel mutations at the locus of interest. The Niakan lab at the Crick is interested in the identification of the factors and pathways that allow a zygote or a fertilized egg to develop into a blastocyst prior to implantation. And CRISPR is a very important tool to perform these kind of functional studies in this specific context. So Nora Fogarty, who was a postdoc at the lab, but who is now a group leader at King's College London, used CRISPR-Cas9 to study the function of the pluripotency factor OCT4 in human embryogenesis. OCT4 is encoded by the PAU5F1 gene. So Nora designed a single guide RNA to target its PAU domain. And this is the functional domain of the OCT4 protein, the one that allows it to bind DNA. Then pronuclear zygotes were microinjected with the CRISPR-Cas9 protein and the single guide RNA. And based on genotyping of single cells at the cleavage stage and at the blastocyst stage, Nora observed high rates of editing. So that means that these samples were OCT4 null with stereotypic 2 base pair indels. And also there were practically no off-target effects. So the experiment was successful. However, a considerable number of samples were genotyped as wild type. That means that there was no evidence or no sign of the CRISPR-induced indels. And these cases were originally interpreted as unsuccessful targeting events. But then a series of publications started to emerge reporting that CRISPR can induce large deletions and complex genomic rearrangements in different biological contexts, like mouse embryonic stem cells, early human embryos, and also P53 competent and P53 deficient cancer cell lines. These events are often missed because genotyping after CRISPR usually involves the PCR amplification of a short genomic fragment around the cut site. So suppose that we want to use CRISPR to create a double-strand break here, but only the paternal allele is affected. And to genotype our sample, we designed primers to amplify and sequence a 500 base pair fragment with the cut site in the middle. Now, any unintended CRISPR-induced large deletion larger than 250 base pair on either side of the cut site can induce a loss of heterozygosity event. We call it loss of heterozygosity or LOH event because this heterozygosity at the site is lost. It can abolish one or both primer binding sites. And since only one of the alleles is amplified, it can cause the impression that CRISPR didn't work or that the maternal allele in this example was used as a template to correct for the double-strand break, when in fact CRISPR generated more damage than expected. Therefore, we decided to revisit the data and material produced in the original OC4 study, which were derived from single cells or from clusters of 2 to 5 cells biopsied from the trophectoderm, which is the tissue that will give rise to the placenta. And we developed computational pipelines to answer the following questions. Can unintended CRISPR-Cas9 loss of heterozygosity occur in edited early human embryos? If that is the case, how frequent and how big are these events? And importantly, do these unintended mutations undermine the findings from our original OC4 study? So let me start with the low-pass whole genome sequencing data. They were produced with the SurePlex kit. Basically, this kit was used to amplify total DNA from, as I said, single cells or trophectoderm biopsies. This kit is commonly used actually in fertilization clinics for pre-implantation genetic screenings. After preprocessing and quality control of the samples, we ended up using 65 single cells or biopsies, 25 OC4 targeted samples, 16 Cas9 controls, and 24 uninjected controls. And we built copy number profiles using these software packages. We use the generated profiles to investigate the presence of chromosomal abnormalities with a focus on chromosome 6, which harbors the PAU5F1 locus on the P arm of the chromosome. So what you're seeing here are the 22 chromosomes with X and Y, and here you have the copy number in log scale. So basically, if you are at 0, it means that you have 2 out of 2 copies of the chromosomes. If you are at the log 2 of 3/2, it means that you have 3 out of 2 copies, so you have a chromosomal gain. And if you are at -1, it means that you have only 1 out of 2 copies. So while most of the samples that we analyzed had 2 copies of chromosome 6 with no obvious cytogenetic aberrations, we found that 8 out of the 25 targeted samples had evidence of chromosomal abnormalities on chromosome 6. So 4 of the samples, or 16%, had either, because these were actually examples of the normal cases. But yeah, so I was saying 4 out of the 25 had chromosomal gains or losses directly adjacent to the PAU5F1 locus, so actually directly adjacent to where the CRISPR-Cas9 editing occurred. And then there were other 4 samples that showed evidence of a whole chromosome gain, so a whole, an additional chromosome 6, if you want. And this proportion of samples with the chromosomal aberrations was significantly different than in Cas9 controls, and of course significantly different than in uninjected controls which didn't show any chromosomal aberration. Moreover, these aneuploidies are specific to the P arm of chromosome 6 because chromosomes 5 and 7, which are the closest in size to chromosome 6, do show whole chromosome abnormalities, but no P arm segmental losses or gains. So So this is how we use the low-pass whole genome sequencing data, but the copy number profiles that we built can only provide a coarse-grained karyotype analysis. To independently investigate the prevalence of LOH events at finer resolution, we perform targeted deep sequencing with the genomic DNA extracted and amplified with the Reproduction kit. After preprocessing and quality control, we kept 52 samples for this analysis, and we had to implement a pipeline that was specifically adjusted for deep sequencing data from single-cell amplified DNA. So the Reproduction kit is very common in single-cell applications due to the proofreading activity of the 529 multiple displacement amplification polymerase, but this comes at the expense of high preferential amplification rates. And that's why our variant calling pipeline has a step to control for allele over amplification biases. To produce the necessary data for this analysis, we designed PCR primers to amplify 15 fragments spanning a region of 20 kb, including the PAU5F1 locus and the cut site. Then we sequenced the PCR fragments with Illumina MiSeq, and we took advantage of the high coverage at each of the fragments to call SNPs. So the bicolor bars here represent heterozygous SNPs, and the red bars represent homozygous SNPs. And this allowed us to identify samples with putative LOH events, so cases in which heterozygous variants that are indicative of the contribution from both parental alleles cannot be confidently called, especially in the amplicons flanking the cut site directly. And we call these putative LOH events because we don't have the parental genotype of the analyzed samples as a reference. The reason being that the embryos that we analyzed were anonymously donated as surplus to infertility treatment. This means that we cannot exclude the possibility that these regions of homozygosity were inherited. So what we decided to do to distinguish between inherited homozygosity and potentially CRISPR-induced homozygosity was to use the presence of heterozygous SNPs in different cells from the same embryo. And we use this as evidence that in this sample, the heterozygosity was lost. So this data led to the identification of 4 different patterns. In samples without loss of heterozygosity, we were able to call heterozygous SNPs in multiple fragments or in multiple amplified fragments. Then in cases with putative loss of heterozygosity at the locus, we found heterozygous SNPs in the amplicons overlapping with exons 1 and 5 of the PAU5F1 gene, and then homozygous SNPs in between. And this could represent CRISPR-induced large deletions of around 4 kb. Now, interestingly, this was the most common pattern in Cas9 control samples, which may indicate the possibility of technical issues due to sequencing or overamplification of one parental allele. Then we found bookended samples which have 2 heterozygous SNPs flanking the cut site, but in amplicons outside of the PAU5F1 locus. And these cases could represent deletions of lengths between 7 and 12 kb. And finally, in open-ended samples, it was not possible to find heterozygous SNPs in any of the fragments. This was the most common pattern in targeted samples. And this pattern could represent deletions of at least 20 kb, which is the size of the region that we explore. Now, these results must be interpreted with caution given the presence of LOH events in the control samples. So as I said, we implemented a step to control for allele amplification biases, but we estimated the level of corrections from the data because, as I said, we lacked the parental genotypes as a reference to choose a more informed cutoff. So we cannot exclude the possibility that the analyzed single cells inherited a homozygous genotype in the explored region, and that events like this one are not associated with CRISPR editing. Nevertheless, the fact that there is a significant proportion of CRISPR-Cas9 targeted samples with the largest LOH patterns is notable. And finally, some of the samples from our previous study were processed with the GNT-seq protocol, which allows one to obtain both genomic and transcriptomic data from single cells. We use the transcriptomic data for two purposes. First, to generate digital karyotypes, like the ones we build with the low-pass whole genome sequencing data, but based on expressed genes or on expressed SNPs. For the sake of time, I will not talk about these results, but I will talk about the second purpose, which was to check if the on-target complexity observed in these datasets may encompass the mutations of genes adjacent or telomeric to PAF5F1 that could complicate the use of CRISPR-Cas9 to understand gene function in human development or any other context. So when we focus on the chromosome location of transcripts, our analysis indicated that the proportion of differentially expressed genes in OCT4 null versus Cas9 controls was not biased to chromosome 6. So basically, each one of these points represents a gene that is differentially expressed because it is about these reference lines. And we mark in red chromosome 6, but it is not that we have a lot of 6s here, right? So the other chromosomes are also represented. And we observed the same basically when we looked at differentially expressed genes for the same contrast, but they were directly adjacent to, or actually telomeric to the cut site, right? So we have a relatively similar proportion of genes here than here, when we normalize by gene length, of course. So these results suggest that the transcriptional differences observed as a consequence of PAF5F1 targeting are not confounded mutations of genes adjacent to the on-target locus. So in all, we found that unexpected on-target complexity following CRISPR-Cas9 genome editing can occur in early human embryos. Our data suggests that around 16% of samples exhibit segmental losses or gains adjacent to the PAF5F1 locus, and loss of heterozygosity events that can span anything from 4 to at least 20 kb in length. However, the discrete on-target events predominate, which may explain why we didn't detect any preferential misexpression of genes on chromosome 6 or upstream PAF5F1, and means that the interpretation of functional studies in human embryos is not confounded by these LOH events. But the material that we used wasn't ideal. So as I said, we use shallow sequencing data, multiple displacement amplified DNA, we lack parental genotypes of our samples. So really to fully elucidate the damage that can occur at the on-target side after CRISPR will require the development of a pipeline that combines enrichment of the region of interest and deep long-read sequencing, most likely. In any case, our work strongly underscores that there is much more to be learned about CRISPR-Cas9 and how cells resolve the damage that occurs following genome editing. And hopefully these kind of studies will inform the debate about the clinical use of CRISPR-Cas9 and assess its safety in human embryos. So to close, I would like to thank all the members of the NIACAN lab for their support and Kathy for her guidance and supervision. Also many thanks to Jasmine, James, our collaborators in and outside the CRIC core facilities, and also Robin, Alex, Maxime, and Ashka for great discussions and advice. Thank you very much.
Thanks so much, Gregorio. Really, really fascinating work. And I think we're going to start to see more of these things the more we apply the tools to embryos. I think we have so much to learn. And I'm sure you feel the same, that we are just in our infancy when it comes to learning about embryos in general, let alone about how they're going to react to these technologies. Exactly. So it's amazing pioneering work that you're doing. We've had a number of questions. I'll only ask one for now because we're going to open up. But do you think we will be able to uncover more reasons for infertility? It's a multifactorial condition, but do you think that somebody has asked that it will go a certain length towards treating infertility, or do you think it will just aid in our discovery, or both?
No, definitely. I think that, as you said, the more we study let's say, the molecular basis of human development, the more we will learn not only about infertility, but many other things that are important for an embryo to develop. Right? So, and I think that CRISPR is one of those tools that will be key to, understand all of this.
Everything.
Right? Yeah.
It will change everything. Exactly. Great. Amazing, amazing talk. Our final speaker is Dr. John Appleby, who is a medical ethicist, and his research focuses on reproduction and reproductive donation. He will be discussing a great title, CRISPR, IVG, ART, OMG, and he's going to start untangling some of the ethics of radical innovations in assisted reproduction. John, I can't wait to hear your talk.
Thank you so much. And I just want to say I'm delighted to join Sissy, Helen, and Greg for this webinar. And I also want to thank everyone at i3 for having me and for putting this on. You've done tremendous work. It's a privilege. So yes, as Helen said, I'm going to talk a little bit about untangling the ethics of radical innovations in assisted reproduction. And generally here, just a second, minimize something. All right, so overall, what I want to do is touch on a few of the kind of radical and new assisted reproductive technologies that are emerging, both CRISPR and a few others, and then say a few things about some of the ethical themes that intersect with them. The context is that society is currently experiencing the emergence of a wave of what I have termed radical innovations in assisted reproduction. And these radical innovations build on past research, but they also unlock completely new scientific, medical, and reproductive possibilities. I argue that these possibilities are radically different from what have existed until now. Radical innovations such as CRISPR, in vitro derived gametes, also known as artificial gametes, I'll refer to them as IVGs, and mitochondrial replacement techniques are a few of them, and they raise complicated ethical questions. Some of these questions we are familiar with, such as, how do we consent to a procedure or technology that has never been used before? However, some of these questions we are less familiar with, such as, what are the moral harms and benefits of having children whose recent genetic ancestors were only embryos? So just an overview of the talk. In this presentation, I'm going to discuss how radical innovations such as CRISPR, IVGs, and MRTs, so mitochondrial replacement techniques, are very different technologies. I'm not trying to equivocate them here. They're very different in important ways, but they nevertheless have several ethical themes that are common to all of them. And so I was hoping to highlight some of these themes. And they're not the only themes, but there are a few of the ones that often pop up, and I think they're important. I want to draw your attention to a few of these themes that tend to stand out and become some of the most contentious in the ethical debates that take place globally with arts in general, but also these ones. By no means is this meant to be an exhaustive or conclusive. And the ethical questions that emerge in relation to each technology will naturally differ to some extent. I'm not trying to say they're all the same, and I'm definitely not proposing some sort of grand ethical theory for arts. These are more than anything some observations. So 3 common ethical themes that I want to touch on are identity, and I'm going to say the most about this one. Next, I'll say something about the value of trust, and then finally, I want to talk about weighing the benefits and harms in relation to time. So, for example, how quickly should we move from bench to bedside while maintaining safety when developing radically new innovations? So the first thing I want to do, I just need a little bit of water, is give a tremendously brief and, quite frankly, oversimplified account of some of the science, just because it is some of the first time we're really mentioning IVGs and mitochondrial replacement techniques in this webinar. Helen made brief mention of it. I'm not going to explain CRISPR. I think that's been really well explained. And quite frankly, I'm not even sure my really brief explanations are going to be adequate, but I only have a few minutes to give them. So with in vitro derived gametes, the main method of creating in vitro derived gametes, in the most basic sense, you can use stem cells, and these are derived, depending on the technique, from embryos, somatic cells, or reproductive tissue. And these stem cells would then be used to cultivate gametes in vitro, which then could be used to create embryos. The goal here is to help prospective parents who don't have their own gametes to use, help prospective parents, including same-sex couples, ensure that their children are genetically related to them. It could help solo parents have children who would be, in particular, only genetically related to them. And it could also solve donor shortage problems in some places in the world. So it's an attractive technology, although it's not currently available for use in humans. And then there's mitochondrial replacement techniques. Again, this is a bit tricky to explain concisely in a few seconds, but I'll try to give a very brief overview. So there's a few different techniques out there. In fact, there's more than these three, but these three tend to come up the most. So one's pronuclear transfer, another is maternal spindle transfer, and another is cytoplasmic transfer. So there's acronyms for each one of those. How do they work? Well, mitochondrial replacement techniques are meant to help avoid maternally transmitted mitochondrial DNA disorders. Mitochondrial DNA is maternally inherited. So with PNT, this technique involves taking the nuclear DNA from embryo that has mitochondria that carry harmful mitochondrial DNA disorders in them, mutations, and you take the nuclear DNA from the intending parents that you want to use to create offspring, and you transfer it into an embryo that's been created using a donor egg that contains healthy mitochondria, and then you have a reconstructed embryo, and you can use that to try to have a child. With maternal spindle transfer, same general kind of idea. You're taking the nuclear DNA out of an unhealthy egg, and you are transferring that into a healthy donor egg, and you have a reconstructed egg that you can then fertilize. Cytoplasmic transfer entails taking cytoplasm that contains healthy mitochondria from donor's egg and then transferring that into a recipient egg in order to, I guess, it's not exactly accurate to say, but to kind of dilute the cytoplasm that may have harmful mitochondrial DNA mutations. And so that's generally a grossly oversimplified explanation of the technologies. But the idea is to avoid offspring inheriting or at least experiencing serious mitochondrial DNA diseases and breaking the genetic tie of mitochondrial DNA inheritance from the female progenitor who contributed nuclear DNA. In particular, the inheritance of certain diseases can be avoided in some cases. So the first, now with that basic science out of the way, I can say a little bit about some of the first ethical considerations I wanted to raise. So the first one is identity. Identity comes up a lot in our debates. So how will technologies like CRISPR, MRT, or IVG impact the identity of the people who are created with them? So when I say identity, that can mean a lot of different things, especially to philosophers. But what I'm talking about in this particular case is a person's sense of self. So in the psychological sense of identity, who am I? We know that this sense of identity features prominently in other art-related debates, such as those concerning if and when people should be told that they were donor conceived. For example, if someone finds out later in life that they were donor conceived, they may feel as if they were morally wronged because it harmed their sense of self, of who they are, their identity. So identity comes up a lot in arts debates. And what I want to raise first are two common claims we hear, and then try to make a bit of sense of in relation to them, in relation to kind of all of these technologies. So the first claim or question that's raised is, What if it damages a child's sense of self to be brought into existence as a designer baby? Helen mentioned designer baby. So this language is often used when talking about offspring created using CRISPR. This language, however, I think could be harmful to people created with these technologies, and we need to stop using the term. Ethicists, I will admit, are partially to blame for this. We often use the term in order to create a bit of controversy and draw attention to our work. And I think this is morally irresponsible and something we need to move away from. We need to pay closer attention to how our language could harm the identity of those created with these technologies. So it is significant to people's identity, I think, how we talk about them. Next, we might think about the claim that children brought into existence with these technologies, any of these technologies, will have their sense of self harmed because they feel like subjects of experimentation. So I raised this point because it was a claim that was brought up when the UK was thinking of legalizing mitochondrial replacement techniques. And it's interesting because I'm now seeing it come up again in Australia's debate about legalizing mitochondrial replacement techniques. So I would argue that there's actually no evidence to support this type of speculative claim. The type of experimental lives that critics talk about is characterized often by follow-up on the welfare of children to ensure normal development. And in many ways, this is not much different to the types of regular checkups everyone has, and it's unclear how this would translate into harming a person's identity. But nevertheless, this claim that it would make people subjects of experimentation and hurt their sense of self is something we do see often. Now I want to talk about some issues that directly relate, although they nevertheless pertain to the technology more broadly, but directly relate to each of these technologies. So for CRISPR, we need to potentially think about, in relation to identity, how will gene editing impact a person's sense of self? On one hand, will alleviating genetic disease be viewed as a benefit to one's sense of self after seeing, for example, other members of your family or community suffer from a particular disease? Will parents potentially see this as a benefit that they can confer on their offspring? So I'm removing the burden of you having to identify as someone with X disease, and CRISPR can help me give that to you, and that's going to benefit their identity. Is this something we should think about as breaking one's genetic tie? Multiple people, for example, sharing this genetic disease, so almost like a political or community identity. And maybe instead, other bonds could be strengthened in relation to identity, so people that have shaken off this disease. It's hard to know exactly how this is going to play out, but no doubt CRISPR will likely have an impact on how people think of whether or not they've been benefited or harmed when it comes to their identity. If we think about mitochondrial replacement techniques, we might ask, how will children be conceived, or rather, how will children conceived via mitochondrial replacement techniques respond to media and policy that often labels them as 3-person IVF babies? Again, this is a question of the language we use, and is there a chance that this could harm people later on in the same way that sometimes donor-conceived children claim harm from being referred to as test tube babies? We might also ask, how will children relate to their identity, if at all, to their mitochondrial donors? So will they feel that there's any kind of relation there? That might depend on the different nature of mitochondrial DNA from nuclear DNA. It's hard to know, but how we navigate that might also affect their sense of feeling treated justly in relation to their identity interests. We might also think will it be of significance to their identity that they have a mitochondrial genetic tie to a third progenitor. So we typically might think of people, having up until mitochondrial replacement techniques, just genetic ties to 2 parents. What is adding a third mitochondrial parent? And of course, the word parent's really difficult. That's why I've used the word progenitor. What is that going to mean, and will that have any impact on someone's sense of self? We know that mitochondrial DNA does not necessarily impact on people's personal characteristics and things like that. We don't know yet, though, what impact it's going to have on people's sense of identity. Then finally, IVGs, some identity issues. And I'm going to maybe stretch this a little bit into almost a little bit of the science fiction at the end, because I think it's an interesting thought experiment. So IVGs have become the subject of intense criticism because some argue that there's no reason to use them except to have your own genetically related child. And it's argued that this only serves to further reinforce social attitudes that genetic relatedness is essential to parent-child relationships and the healthy development of a person's identity. And this is generally a narrative that, for example, the donor conception community have been trying to shake off. So IVG could potentially be something that, undoes that good work. I have argued that this is mistaken, and IVGs could be used to, in fact, disrupt the status quo of reinforcing attitudes that genetic relatedness should be valued. And so just hear me out. IVGs can also be used to bring children into existence whose most recent genetic relatives were, in fact, embryos. If we create embryos, we use those stem cells from the embryos to create new gametes, and we create this process several times, we could get what another philosopher named Robert Sparrow has termed genetic orphans, wherein the nearest genetic relatives to any child created with these IVGs would indeed just be embryos. And it would be very hard to identify a recognizable parent figure or clear progenitor that we would relate to in normal genetic relatedness terms that actually lived as a human being. And I think that parents may, if this actually happens, parents may feel that there's a benefit to using IVGs in order to have a child that is related to no one, because it means there are no living donors in the picture that could complicate a child's sense of self. And we do know, despite the fact that I personally don't think having donors in the picture is a problem, we do know that some people that seek assisted reproductive technologies prefer this. So it's a question of taking their preference seriously. They might prefer to just have the child related genetically to no one. So in this sense, the genetic tie really wouldn't matter at all. And I think it kind of turns the whole thing on its head. So therefore, what are the implications for a child's identity if they have no genetic relatives that are human beings? I think that's an interesting question in relation to identity that we've really not thought enough about yet. Now I'm going to talk just very briefly about trust. What is the significance of trust when taking a technology from bench to bedside? First, I think we just need to identify what trust is. So a philosopher named Karen Jones, I think, does a nice job explaining trust in her paper, Trust is an Effective Attitude. So she says, trust is an attitude of optimism that the goodwill and competence of another will extend to cover the domain of our interaction with her, together with the expectation that the one trusted will be directly and favorably moved by the thought that we are counting on her. In other words, people might trust scientists working on CRISPR because they feel that those scientists will recognize their trusting attitude. They'll recognize their trust and reciprocate by acting in a trustworthy way. So it's a relational concept. And I'd also like to highlight that trust is different from reliability. Reliability is based on evidence, a track record. Trust can use reliability as a measure of trustworthiness, but it doesn't depend on it. And I think what the magic of trust is that we can trust on bad evidence. And that's significant because Helen highlighted how things had gone wrong with He Jiankui with respect to CRISPR. And if trust was purely based on evidence, then we might not be able to develop the optimism needed to continue pursuing these technologies and develop trust that they'll be done right next time. Trust matters because we need to cultivate this attitude of optimism in whoever we need to trust our technologies and our actions with those technologies. These technologies, whether it's CRISPR, IVGs, MRTs, or any other radical innovation on assisted reproduction, they're no use to anybody if nobody wants to use them. So the process through which we debate and communicate the science and implications of these radical innovations will likely dictate how much they're trusted. So we need to think about cultivating the trust of several groups when we're bringing these technologies from bench to bedside. We need to think about society as a whole. We need them on board in order to pass laws and permit these technologies to be used. We need doctors and healthcare professionals to trust them. We need them to trust the tech in order to use it on their patients. We need parents to trust them. We need them to feel safe and trust what is being used. And we also, I think we often forget about this, need to have future persons on board. They're not obviously in existence yet, but we want offspring to look back on how they were brought into existence and trust our judgment. And the last ethical point I wanted to raise was this age-old question of how do we weigh up benefit and harm versus time? How do we weigh the speed of introducing a radical innovation versus the possible risks and benefit of doing it quickly or slowly? There's a lot of different arguments that are made. So one of them is that, on one side of the spectrum, every second we waste, we are missing another opportunity to help another person wishing to have a child that this technology could help. We need to hurry. Another perspective on the other side of the spectrum would be, if we rush and something goes wrong, then we're doomed. Then we've doomed not just the technology, but all the top technologies in the pipeline that are like it. So we need to be very, very cautious. And the question is, what's the right answer? What's the right balance between these two, the two sides of the coin? And again, I think trust can be helpful here to bring trust back in. Different parts of the world may seem to be in close competition to get a world first. We've seen that with mitochondrial replacement techniques. We're now seeing it with CRISPR. It's likely we will continue to see this with radical innovations, everyone wanting to get a world first and potentially even be willing to cut some corners to do that. However, at the end of the day, it is trust that is the currency that all of science trades in. So damage to the currency of trust by any single entity or group of entities may then damage the entire scientific field which it is a part. And that is to everyone's detriment. So we need to weigh that up. We need to weigh up the speed of innovation versus harm and benefit. We need to consider what course of action will foster trust in the end product. Now, I think what I've really done here is ask more questions than give answers, but I think we need to approach all of these radical innovations with lots of ethical questions, lots of ethical scrutiny, and pay attention to all of these different ethical dimensions. Thank you very much.
Thank you very much, John. One of the great things about having you here and having this as part of the conversation is that means that we're having this ethical discussion kind of in real time, right? So we have this emerging technology coming through, and we have all these concerns and these questions, and it's nice to be able to have both the scientific avenues and the ethical avenues being discussed at the same time. So thank you very much for that. Related to that, so I have a question for you. So in the case of mitochondrial replacement therapy, one of the big things was a consultation with the public in the UK. They had these wide-ranging conversations at the same time that the science is going on. Do you think that now is the time to start doing that with CRISPR? Has it already started? And how do you envision the roles of various members of the community in that schema?
Was that a question for me?
That was a question for you, John.
Yeah, and I'd like to highlight, I think there's always people that criticize the UK process. Incredibly difficult thing to do. It was wide-ranging, it was detailed, it was lengthy. It was really world-class, and I think a model for everywhere else. And I know other people do use it as a model. So my personal view is that it's always dangerous to start a conversation. I think for scientists, at least their point of view, started too early because it seems like it's less grounded in maybe certainty, the technology, there's more questions. At the same time, we need to start it as soon as possible because I think it does cultivate that trust to make everyone feel like their voice was heard, allow all the evidence to be. There's nothing that I think endangers trust more than to make something feel rushed. That's a really general answer, but I think we could probably we need to start talking about IVGs now in CRISPR now.
I couldn't agree
Great.
more. I think even at the time when people were saying surely it's too early to talk about this, boom, the news hit, and actually, you know, this presumptive idea that, or merely conjecture, actually was very real. So there's no time is too late to start talking about the applications of these therapies. So we're going to go into our Q&A session. We have lots of people and we have lots of questions. So we're really excited to just put a bit of a focus on each of the questions that have been given to each of the speakers, myself included. So Gregorio, I'd love to start with you and ask you one of the questions that was directed. And I think it's a really good question because it asks about whether you think the high losses and gains from CRISPR-Cas9 may be embryo quality related. You know, were the embryo samples obtained from apparently normally developing embryos, or were the experiments performed in spare embryos not suitable for transfer?
Yeah, no, these were embryos that look healthy, and actually, I mean, the important point here is that these embryos were microinjected. I mean, the ones that were targeted were microinjected at the zygote stage, right? So, and one of the findings in Nora's paper was that targeting OCT4 was detrimental for the appropriate development of the embryo. So, but the control embryos, the Cas9 controls, they look healthy. But of course, I mean, these embryos are frozen, then you have to thaw them for the experiments. I mean, of course, that can also have an effect on the quality of the embryo at the end, right? But yeah, I mean, I guess Nora and the others in the lab did the best they could to control for all these factors.
Amazing. It's very difficult when I think our only sources of human embryos are those that are donated, that generally come from possibly women of advanced maternal age. They have been through the freezing process, etc. So yeah, it's I always say that we're probably dealing with the most faulted system there is. You know, I think the technology will be amazing always, and it will continue to advance, but there's actually very little we can do to advance the innate cellular biology of a human embryo, which is actually for the most part destined to fail in a lot of places. So yeah, thanks for that.
Great. How about you, Helen? Let me ask you a question. So this comes again from our Q&A. And the question is, do you think population screening for genetic conditions, such as single gene disorders, number one, are a good thing? And do you think it will become more common as we go forward?
Yes. I mean, I think, truthfully, I think every aspect of our healthcare will become much more tailored, from every individual being sequenced to knowing their pharmacogenomics to any of the tailored therapies will rely on personalized elements. In fact, I actually would consider it almost medical malpractice to not do screening and identification of, you know, resistant mutations that can basically provide a strong confidence that the small molecules will work in a given individual. So we know that there's so much variation within people, and so assuming that everyone will react differently, it breaks all principles when we look at modern pharmacology and pharmacogenomics, that actually, you know, as many as 1 in 10 people will not process or react to coding. So I think we'll see so much more of that, and I think it's an entirely good and feasible thing with the advances in both technologies, but equally the reduction in prices when it comes to sequencing.
Yeah, I think that's an excellent point, right? Personalized medicine, indeed, you know, our session is personalized genetics, are sort of, you know, the next frontier, ultimately. Like you said, tailoring it in such a way that individual people have these options, right? And then a question for the three of you to perhaps discuss is, again, from the Q&A, it's a hypothetical question, but when do you believe that gene editing will happen in everyday clinical practice? So I think this covers all your topics quite well, right? So Gregorio, you have the technical aspects, the ethical aspects, Helen working very much at the frontier of the field. Do you care to speculate?
So we've already seen it being applied to beta thalassemia, sickle cell. The first patients have received doses. We've seen incredible results from those. You know, I think there's been over 30 patients that have had delivery of CRISPR editing components. They have not shown any further. These are patients who were having crises very often, and they're in a much better state. So I think when we start to see, again, we can't compare with normal clinical trials, which require a very long time to prove something's worked. When it works in phase 1, it makes you wonder how can we expedite the process. I think it's going to be rapidly seen, as I mentioned earlier, that this finding method that the guides can do to actually precisely locate a place in the genome doesn't have to be used for genome editing. It can be used to say whether somebody is, and in fact, some of the CRISPR targeting methods have been used for detection of COVID as well. So I think it's going to be a very rapid advancement.
Yeah, I mean, I think that at the embryo stage, yeah, I mean, there is targeting at that stage is difficult, right? And I think it will take a lot of time to learn how the embryo, resolves these double-strand breaks and so on. I think that probably, I mean, the genome editing technique that will be being applied to embryos probably will be won't be CRISPR, right? Probably will be something else. I don't know if it will be prime editing or any other more advanced technology, but I mean, definitely it will be there, right? But I mean, for me, I think it's difficult to say it will be in X amount of years because of what I've seen, right? So the data shows that there are still things that we don't understand. So I think that a lot of basic science need to be put into into this field to really understand what's going on and make 100% sure that what we're doing is safe.
The only thing I'll add to that is that I think one of the things that will need to perhaps start happening, continue happening in relation to the technology is that there'll need to be a lot more public education of what the implications are, how it actually works, how certain the science is, because ultimately I think that will, at least in regulatory areas like the UK, it depends on where it is, that is an important ingredient before it actually becomes a mainstream reality for clinical use. And I think that's probably been one of the underpinning aspects of success of other technologies that are, quite new, like mitochondrial replacement techniques, successful public education to some extent.
Yeah, I think we can never have enough public education. At the end of the day, I think most of the people that are readily, involved actually become experts in this, and those are the parents of people with rare diseases and children with rare diseases for sure. They become experts overnight. Gregorio, we have another question for you here, and that is: do you think that there is a time when loss of heterozygosity would actually be desirable?
That's a good question. Yeah, well, there is actually a paper by Dieter Egli who actually, I mean, after CRISPR in embryos observed more or less the same events that we saw. So segmental losses of gains directly adjacent to the CRISPR editing site. And he proposed in the paper that probably you can use, I mean, take advantage of the fact that CRISPR is affecting the chromosome to, for example, get rid of trisomies. Right, but yeah, I mean, this is something that again we have to do a lot and learn a lot to see if this is possible or not. I mean, the problem with loss of heterozygosity is that usually they cause more problems than benefits. Right? Actually, this is like one of the mechanisms that cancer uses to spread and develop. Right, and I mean, and loss of heterozygosity doesn't necessarily mean like a complete homozygosity in a region, right? It can be just a point mutation, and that changes the heterozygosity you have at the site. But I mean, on the other hand, there is a group suggesting that after CRISPR, the embryo uses a maternal allele to correct for the double-strand break, right? And in this case, using the healthy allele to correct for example, a problematic gene. And I mean, if this is actually the case, and this is yet to be proven, then this could be a scenario in which loss of heterozygosity is desirable, right? So actually, you want this mechanism of gene conversion to, you want to exploit it to actually correct the gene. But again, so this is something that we don't know yet.
I think there's a lot of information still, or more experiments to be done to really look into, you know, that phenomenon of maternal, you know, the reliance on the maternal transcript. So it would be great if it was. I think there's a number of experiments still to be done in order to actually really verify that, for sure.
Yeah. I mean, it's really challenging, right, to test for this. And I think that, for example, long read sequencing and other technologies will be instrumental to learn what's going on.
Yes, for sure. Yeah, exactly. So thank you to Gulam, Zach, Kalal, Rena, I hope I haven't mangled your names, and Rachel. Thank you to everyone who has asked questions so far. We're trying to get through to everybody's. We really appreciate these questions that you have put in.
Agreed. I mean, Greg, while you're on the topic, we have a question from Gulam Murtaza, who is asking, can genome direct correction during the cell cycle remove gene editing, as after transfer, many more divisions and correction points come along? So what happens after it's happened? Is it permanent? Is there risk it's going to pass out, or how do we deal with this?
Well, this is also really difficult to answer, right? Because yeah, especially as Helen mentioned, you have a lot of mosaicism in the embryo. So, I mean, some cells do inherit the edited alleles, but some don't. So, yeah, I mean, I don't know if it is permanent, as you said, or not. I mean, we have to do experiments and investigate it.
Yeah. Yeah. Check back in a little while once you have a few more experiments done, a little bit more.
We'll be keeping our eye on you. John, I have another question, if you don't mind, from Philip Chan, who says, injecting sperm via ICSI includes transferring paternal mitochondria to the oocyte. It seems the paternal mitochondria degrade with each cell division. Shouldn't we be less concerned with MRT?
It's possible. I mean, maybe we should be. There's people definitely that go both sides on this. There's a whole slew of concerns about exactly how concerned we should be. Haplotype matching. There's all kinds of things. But the fact of the matter is that maybe I think one of the most important debates, I guess what I'm trying to say, is putting any kind of safeguards in place so that we really understand all the implications of using these technologies for the first time. I'll try not to make this too long-winded, but when the UK was talking about licensing mitochondrial replacement techniques, the early versions of the license for using these technologies that they trialed with, made public, included no long-term follow-up for the actual technologies. It was not a requirement of the license. In terms of the license they made public at their consultations. It was later added in, so now it is a requirement. But having something like long-term follow-up is an essential aspect of trying to determine exactly whether or not some of our worries are things that we should still be worried about or not. Maybe it amounts to nothing. I think those kinds of safeguards are essential to ultimately answer those kinds of questions.
Couldn't agree more.
How about, and again, another question for the three of you. So this is from Anya Skevin, who asks, would IVGs lead to a reduced genetic pool? And how can we also look at that from a bioethics point of view?
Could you replace, could you repeat that one again?
How would IVGs and in vitro gametogenesis in general lead to a reduced genetic pool? And how can we look at that from the bioethics point of view?
That's a tricky one. There would obviously, so people have raised concerns about in vitro eugenics, that this could be combined with CRISPR and there could be a perfect storm and all kinds of things could go terribly wrong. And so there's definitely every possible scenario that people have brought to light about how concerned to be about some of this. But then there's the reality of the fact that we do all kinds of things and we just regulate them properly and it's fine. I suppose it really depends on how they're ultimately used, but it's not something I'm particularly concerned about, reducing the gene pool through IVG, but there might be people out there that are. I guess this is why we need to be having these kinds of discussions to kind of dispel any concerns. But maybe, Sissy, maybe you also have views on that.
I think so. Partly I agree with you. So yeah, it's part of the reason that you have the conversation is that the ethics need to match up to what you're doing, right? So you made a great point about licensing, and I think Helen brought it up in her talk as well. There's several technologies related to fertilization that would have been unthinkable, 50 years ago. But the question is, we've had a conversation about IVF, moving from test tube babies to a very widely taken up technique now. Similar conversations about ICSI, similar conversations even about licensing CRISPR. So I don't think it's unfeasible to think that the conversations will move in a good direction.
We hope so. We are here to ensure that the conversations go in the right direction, and hopefully we are able to come up with some answers. I always strongly feel that the UK is one place, but the rest of the world thing, you can legislate in one area, and it doesn't mean that somewhere else in the world they're going to abide by the same rules. And there are those who would argue that you may fall behind by dwelling too long on the ethical arguments rather than the real world scenarios. So with that, I think we can probably start to wrap up. Bear in mind that if you send in your questions and we haven't gotten to them, we will try and get back to them at another stage, or you can contact us or message us to try and answer some of your questions. Thank you, everybody, for taking the time to participate in the session and in this discussion. I particularly like to thank the i3 speakers and my co-host Sissy, John, Gregorio. Thank you to the organizing committee also of this initiative, who have worked tirelessly behind the scenes to rally these these troops to play ball at the right time. So thank you to Jacques Cohen, Peter Nagy, Thomas Elliott, Shaista Sadruddin, Giles Palmer, Marianne says, sorry, I've said that wrong. And today's Q&A team were Marianne and also Anna McLaughlin. So thank you very, very much.
And thank you very much, Helen, both for co-moderating and for your great talk. Thank you to all the attendees who joined our session today, and the session and the presentations will be available on the website soon. It's important to point out that the next session on i3 is from the journal Reproductive Biomedicine Online with Chief Editor Bart Fauser and Emeritus Chief Editor Jack Cohen, which for the first time is going live with interviews, discussions, and showcase articles. So if you join i3 next Tuesday at the regular time, 8 p.m. GMT, 9 p.m. Central European, 3 p.m. Eastern if you're in the US, and we'll be happy to have you. And if you stick around now, we have a short closing video, and thank you very much again for joining.
Hi there, my name is Savannah, and I'm currently completing my Master's in Reproductive and Developmental Biology, and I am an ambassador for i3. If you've enjoyed today's session, please do give us a like and a follow on Facebook, Instagram, Twitter, and LinkedIn. Our social platforms are regularly updated and will provide you with all the information on future sessions, which you can share with your friends. We encourage people to go on our website at ivfmeetings.com, where you can view all the previously recorded sessions, leave a review, suggest future content, and you may even be in for winning a prize. Thank you so much for listening, and we look forward to seeing you in the next session. Goodbye.